Good afternoon, everyone. Today I'm going to talk about our work from past 2 years. It's an open source framework for building and evaluating transcriptomic foundation models, and it's called BMFM RNA. Please scan it, like to see like all of our papers, all of the posters. We were like 10 to 12 posters here, and also we are giving like 2 to 3 talks like from our team. So please go meet them and learn from them and what we are doing today. So coming to the transcriptomic landscape, high throughput sequencing has transformed transcriptomics by generating massive amounts of data. These data sets offer rich potential to decode like cellular behavior and also the regulatory mechanisms. So they lay the foundation for building, so these data sets, for building the transcriptomic foundation models that generalize across tasks and cell types. Like, so we get like data sets which are smaller, which are specific to tissue. But like here, like, so there are efforts started like to bring all these data sets together, and also can we train like the foundation models. So the current foundation models that started recently, like SCBERT, SCGPT, GeneFormer, so they all like propose like a different technique to build these foundation models, specifically where we have, large amounts of data that is coming from different sources. And also the other thing is like, there are batch effects coming from each of these data sets. So each and every method like proposes like different approach, and we don't know like which one is the best, which one can actually do the differentiation, like especially to decode cellular behavior and regulatory mechanisms. So what we did like in last 2 years, like the effort is to bring together all these approaches and provide like a common framework, like where people can actually experiment with different settings to see like which is the best setting that they can use to train the foundation models. So this is an open source modular platform to unify diverse transcriptomic foundation models, training, and as well as the fine tuning. So this is part of like BMFM multi-omic. The GitHub link is here. So this one, we actually have both for DNA and RNA. So we have implementer for DNA models, DNA foundation models, and also here, like we also have like RNA foundation models. So it enables rapid prototyping and evaluating of new models, new architectures, and it comes with like different training objectives, different data processing strategies. So our goal is to promote reproducibility and systematic benchmarking. So this is our overall pipeline. So like, each has details, like, how do we load the data, what kind of quality controls we use, and how do we select the data, like, different kinds of sampling strategies. Like, do we really need like, 50 million cells? Or do we just need like 1 million cells to train these foundation models? That's a big question that we also answer through this platform. So what kind of transformations we allow? And again, like, we build on top of like existing open source libraries. Like for example, ScanPy, LibData, Hugging Face, PyTorch Lightning, ClearML, Captum for, if you want to do like gene attribution. So there are all these different libraries that we brought in here together to actually build this foundational model framework for transcriptomics. And also, like, we introduced a new technique with this paper, and the papers are open source, like, it's in archive now. So the technique is like whole cell expression decoder, which actually captures like global expression patterns using an autoencoder like CLS bottleneck. So it has like 2 losses. That's what we did in this approach, especially like, after experimenting with these different approaches, we learned having, especially with the cells and the cells, like, where you have like genes and expressions, which has like multiple fields. And at the same time, they are not continuous. Like, where you may have seen like foundation models for text, like where you have a sequential data. So we use like 2 different losses. One is like to say whether this gene is expressed or not. And at the same time, we also want to see like, how much is the expression that is in the cell, in that particular cell. So that's our architecture. And here are our zero-shot results. So especially the interesting part here is we used a technique to sample from the data. Again, like a very simple technique. So it's cell type aware, because these data sets from Cell by Gene has like 50 million cells, and they have cell types already annotated. We just did a round robin sampling from this data set, and the number of cells we used is just 1%. So with 1% of the cells, which is less than 1 million cells, we were able to replicate existing foundation models like SCGPT. So as you can see the comparison, right? So with just 10% of the data, like especially in the third column, we were able to like significantly outperform on zero shot cell type annotation. So do we really need this much amount of data? Do we need this big foundation models that can take like a lot of this data? So that's like, we are inviting the community to experiment, collaborate with us, come join us, like talk to us. Like, we were like 10 to 15 people here traveling from different countries representing IBM research. So yeah, thank you.